Okay, then let's continue with this class before the madness.
Tired, no?
Yeah. Thank you for
So, so this is the overall outline of the whole fMRI introduction. So we did introduction overview. We talked about what we can talk about in terms of MR signal generation and static contrast. Now we finish with metabolism, hemodynamics, and Bolt signal, and then the next thing is to talk about spatial temporal properties of the Bolt signal, so that you get a kind of an idea about its spatial and temporal properties. And we are basically then at the half of this course, and then later on we will also continue with fMRI data analysis in classifiers. Spatial temporal properties of the Bolt signal. And there are a couple of things in this section. So first of all, the spatial resolution, temporal resolution, which is fairly clear. Then one thing we need to discuss are the linearity properties, because they underlie all these analyses, especially in the context of GLM, which, if you do a classifier, is I think these days then used in classification studies to do the classification on the beta parameters, because they filled out some noise. So GLM is always there, and classifiers pretty. Anyway, don't want to talk about classifiers now, but I heard this in the break all the time. Signal and noise, sources of noise, and in the end, to discuss a little bit whether more field strength always helps, which is not the case, as we'll see. So first, spatial resolution. So the question is, of course, what is a reasonable spatial resolution in the first place? What is the resolution that one can achieve in terms of fMRI? And what's going on? So this is an overview about the spatial scales in the human brain. So starting from kind of the cellular, even subcellular level of ion channels, over synapses in terms of millimeter neuronal cell bodies, cortical columns, so these units of cortex that kind of have the same function on the millimeter scale, to the more macroscopic gyri that kind of have a centimeter extension, Boltzmann areas, which kind of have a centimeter square extension. If you think about this Boltzmann map that I guess you've seen all, where you have all these different cytotectonic architectures, and then of course the brain, which is cut on the 10 millimeter scale. And with in terms of the spatial resolution of the imaging process, definitely the voxel sizes are usually around, let's say 2. So depends really on what people do, but for a common cognitive neuroscience study, voxel sizes are around 2 millimeters, so it's kind of at the level of cortical columns and gyri. However, voxel sizes are not everything. This is an important thing about this part of the course. However, of course, voxel sizes are the most immediate thing that you think of when you think about the spatial resolution of fMRI. Like I said, typical voxel sizes, I guess in cognitive neuroscience studies at least, are of this size, so 2.5 millimeter. Can be smaller, can also be non-isotropic, so be specifically in the slice thickness be a little bit larger, but that's kind of the ballpark for most common studies. And then, of course, they are determined by the field of view, so depends on how much of the brain you want to image, how big they are, and how many values you acquire. If you do slice-based imaging, that essentially depends on how strong your gradients are and how fast you want to do the imaging, because as we will see when we talk about the imaging aspects of MR later on, we will see that what is happening there is that's the spatial frequency decomposition, and that depends on the strength of the gradients and how many values you get per slice. So the in-plane resolution depends on how your whole setup is. And then, of course, it also the voxel size depends on not only in the XY plane how big things are, but also how thick the slice is, which depends on how many slices you want to acquire and how specific you can fiddle with the Lamour frequency. So this is kind of basic. Smaller voxel size require longer volume acquisition times because you usually need to go go longer because there's a coupling between the time of the imaging process and the highest frequencies that you can encode, which we will see in more detail in January. So on the one hand, if you want to go to smaller voxels, you need more time, which means that your temporal resolution is going down. If you use larger voxels, then of course what you induce is larger, what is called partial volume effects, which essentially just means that within one imaging unit of a voxel you have multiple tissue compartments which have different properties. So this is all relatively straightforward and is something that you can actually fiddle with, so you can define a voxel size. It's not the whole thing about spatial resolution, because this is just the physical resolution of the imaging process. This shows what basically the idea of a partial volume effect, that you have in a given voxel, you have multiple tissue types resulting in a mixed signal from here CSF, so spinal fluid. Here you have some white matter, here you have the cortex, and then you have differently strong vascularized aspects of the cortex, which also then might give differently strong bold signals. And all of this, in terms of your gray level image, is condensed to a single value. Yeah? So this means that if you have very large voxels, it's quite clear. If you have one voxel only that covers the whole brain, then of course you have all signals from all different brain areas. If you go to smaller voxels, of course, you get at least physically a higher resolution. The most important thing about spatial resolution from my perspective is that one thing is the physical resolution that you can achieve with your imaging method. The other thing is the biological resolution, or the effective resolution that the bold signal allows you to have, which of course depends on many unspecified features, so that's really, there's not really a way to quantify that. So if you are thinking, for example, that you want to distinguish two neural tissues, one doing something for faces and the other thing for houses, if these neural populations would be completely intermingled, then it's really hard to dissociate anything in terms of bold signals, because you wouldn't think that there's any spatial specialization in the first place. If there's less functional specialization and in anatomical setting, then meaning that at least at the neural level, kind of the biological resolution and the imaging resolution, they kind of match, so that you with your millimeter voxel resolution and the organization of the brain, you might have a match. Of course, that's completely not a given, right? So there are areas that are more or less specialized for something, but not for everything. We will come to these conceptual issues when we talk about experimental design. The next step, one level above the neural specialization spatially, is of course the vascularization, and how neural effects translate into differences in vascular regulation. So what is shown here is an electrode which is actually used to excite a region of cortex in a slice. So that's not a slice in terms of from an eye, but a slice in a petri dish. So that's an in vitro experiment. And what they measured at the same time, I think it's in a petri dish, might also be in an animal, can also be an animal. but definitely not an fMRI experiment or something. What they did is excite a certain area of neural tissue is not shown here, and measure the changes induced by this in terms of arteriole dilatation in the vicinity of this. So basically to figure out how much is the spread of this neural activation in terms of the vascular system. And what they find is that if they are like 38.7 micrometer, it's fairly small, away from where they stimulated, they still find a dilatation of 8.7%. So, and if they are very close to it, they find a stronger dilatation, for example, here of 31%. So what this shows is that if you have neural activity at a very precise neural area, then you get arterial dilatation induced by these metabolic processes that we discussed, and also not discussed because they are unclear. You get that not only here where you have the electrode, but also further. Now I know what I got wrong. So this is millimeter. So sorry, I was confused. This is millimeter. This is the dilatation, I think. This is the dilatation. That makes more sense with the micrometer. So this is how much the thing dilates. So does it? I think making sense what these numbers are. So in the end, what these numbers are is what the Hurdle caption says what these numbers are. So have a look there. I think this might, so what makes sense would be if this 38 micrometer is the radius maybe of this vessel. So that would be larger if you're further away from it. Here you are, however, are 2 millimeters away. You have a vessel that has this diameter or radius. And it changes, and this is definitely what I remember, it changes by this percentage. Yeah. And my point of this whole thing, what I take from is that you don't only have a change in dilatation exactly here, but you also have it in some millimeter further away, meaning that the Bolt signal, if you would take this whole thing as the Bolt signal, changes everywhere here because you have dilatation everywhere. Yeah? It's a quantitative, it's an attempt to quantify a little bit what's going on, but yeah, it's not that easy. The important thing is that you might have, you have essentially a spread. You have a point spread. I think Nikos and people always talk about the point spread function of the Bolt signal, in the sense that if you ideally induce neural activity only on one side, how much of this point activation neural activity is spread out, and you also measure. And this is really, so I can only say, so what I only dare to say is that definitely the spatial resolution is not only the voxel resolution. It depends on the neural architecture, it depends on the neurovascular coupling, and it depends on the vascular coupling. And this, of course, depends on where you are in the brain. So to really put a number onto it, it's tricky. And then you could also say it also depends on the data processing. So a lot of things that happen in fMRI is also smoothing. So you smooth the observed data, which induces even then bigger activation. So yeah. Spatial resolution is tricky. The spatial resolution that you have in terms of voxels, it's not necessarily what you get in terms of functional activation. So typical things that you see in cognitive neuroscience studies, I would think, is that you do an fMRI study and your blobs that you have after all the data analysis are around 3 to 4 centimeters in diameter, that you really believe, that you think, okay, this is something. If they're really small, then and they are kind of everywhere, then you don't, and they are usually then also quite weak, then you don't quite believe it. So yeah, I think so 3 to 4 millimeter blobs, that's what you get out of fMRI in the end. However, having said that, for cognitive neuroscience studies, you can also, under certain conditions, definitely, and under certain imaging protocols and under certain biological neurovascular settings, get something more. And this is an example, the first example for today, which is the ocular dominant columns in visual cortex. So this is a little bit older. So I think this is early 2000s, where I think the Minnesota group has done these things. So this is human fMRI. However, that might be 40, maybe it's even 70. Maybe it's 3T, I don't remember, but it's definitely done by a group who is really good in magnetic resonance imaging. So this is not in terms of the physics. So this is dedicated work, right? So this is by people who dedicate themselves to the development of MR methods. And, sorry, a little bit dry here. And they acquired data from a human participant, and they acquired a lot of data, and this human participant was really devoted to the whole project, so that also hold very still. I think this was actually done with a bite bar, so that there was no movement beyond the blood movement, but no movement inside the scanner. And they used really high imaging of visual cortex and presented left and right visual stimuli, checkerboard most likely, and then you have parts of primary visual cortex that are primarily active for one eye and the other one for the other eye. So maybe they actually presented not left and right hemisphere, but to one eye and to the other eye, which you can do, of course, if you separate these things. The finding here is that you can actually recover what is known from monkey invasive electrophysiology. You can recover ocular dominance columns, so areas of V1 which have kind of like a 1 or 2 millimeter diameter with fMRI. So this is what shows the raw data, and then the blue is for one eye, the other one is for the other eye, and if one does some smoothing on it, it looks like this, and one sees still this pattern. And they also show that this is reproducible in the same participant, of course under the same conditions. You see it also in the second session. So this shows that if you push it, if you invest into optimizing the MR imaging process, if you use a biological phenomenon that actually exists, so you need these kind of ocular dominance columns to do something like this, and you also need to be lucky enough that the vascularization is so high and so specific and high that this is also reflected, then one can push it to this level. That doesn't mean that you should now expect from every cognitive neuroimaging study that you see this everywhere in the brain, because not everywhere in the brain you have ocular dominance columns or any kind of this neural structure that allows this. Also, you might have an overall other sequence and so on. Yeah, but one can push it to this. And questions about this? Yeah.
About the slide before, actually, wouldn't be the fact that there is some activation in also in a little bit distance of the activated site? Wouldn't that be a reason for controlling, statistical data to control for activation in adjustment boxes?
Yeah, but this you could. So you mean that if you have. So you mean that if you have a voxel that is activated and then you have a voxel that is next to it is also activated, that you downweight the adjacent voxel. You could do something like that. It would, of course, rest on a model, and you would then need to quantify how much you want to downweigh this other voxel based on measurements that suggest what you should do. And then, of course, you also have the problem that you don't know where things happen, and you don't know which is the seed voxel kind of where you should do that. So I don't think that anyone does that. If you in some way can determine which voxel you really believe and which voxel you think is covariation, then that can be done. I mean, in a way, what you observe in real data is kind of a activation that, of course, is most strongly in a certain area and then levels off toward it, towards the edge of it. And if you then, because of these kind of measurements, you say, okay, the border voxels, they are most likely only neurovascular. You can do it. You just need to convince your reviewer and first your PhD or project supervisor. If you're not a professor, if you're a professor, you just need to either do it and publish it and convince your reviewer, or if you need the money for it, convince the funding agency that your model by which you're doing this is valid, and that these data that come from a single study are the... So, yes, it's a good idea, but it's not done actually. Other thing is, I thought, actually what I thought your question was going into is that, because this can also be said about this, if you induce neural activity here and you observe vascular activity here, of course, if you induce neural activity here, neural activity will also spread. And then the question is in how far this vascular activity in terms of dilatation here is due to this neural activity at this origin or neural activity here, which you also need to measure. And this is something one can, in a way, address a little bit with combined TMS fMRI, because there you induce neural activity and you might have a good idea how this neural activity spreads, but this is also really demanding yeah, not something that is easily done. Good, so this was the early 2000 example, and now, as a special treat, I give another example about spatial resolution that is just from last month, or this month maybe. It's on bioRxiv, and it's new work by Peter Bandettini, and cognitive neuroscientists in this department like it, so it was suggested to do a journal club on it. It's new finding in terms of layer-specific fMRI in a cognitive neuroscience domain. So over the last couple of years, there have been not increasingly many, but a couple of layer-specific fMRI papers that say we find that so we use 7 Tesla fMRI, and with this 7 Tesla fMRI and or specific sequences to do fMRI, not only bold but also a new sequence which is called VASO, which not only looks at bold but also the cerebral blood volume. We can do something where we can distinguish different layers of the cortex. So you might remember, I hope you remember, that cortex has different layers, and these layers also have different functions. So does anyone remember input or output layers of the cortex from your neuroanatomy? You all have seen that the cortex has different layers, right? Good. Okay, now let's keep it at that. Input and output layers, I think output is, we will see it on the next slide. I think output is 5 and input is 4 or something like that. It's just numbers, but we will see it on the next slide. We're talking about these kind of layers in cortex, which now people start to investigate with fMRI and different functional properties. However, there are very few of these studies so far, hence if there's a new study, it's all like, hey, there's a new study, it's awesome, but you should also take them with a grain of salt, so because it's often kind of these early studies, as you might know, might, depending on how they are done and so on, they may be easily or not that easily reproducible. So yeah. So anyway, I only believe the facts that I have reproduced myself anyway, so maybe I'm too critical, but in any case. And now this is about fMRI layer specific, and we will see pictures of this layer specific in a second, but it's fMRI in an explicitly cognitive domain, and this is what people here like, because it's not kind of vision, ocular dominance columns, something like that, but it's really cognitive. So it's a working memory task, and what people were shown are strings of letters, random letters, but in their paper they use these random thing, pixel, which makes me think of pixel a lot, but that's supposed to represent a random, non-meaningful sequence of letters. And so they were presented with them, of course, not only pixel, but also voxel and fMRI, so random letters. And they had to do two things with them: either alphabetize them, so manipulate them, or just maintain them. So in one task was to, if you have this PXEDL, you would have to think, so first you need to remember, so you're only shown this for 2.5 seconds, and then you need to remember PXEDL. Of course it helps that it's almost pixel, but so it's PXEDL, and then you have to put them into alphabetical order, so A, B, C, D, E, so D, E, and then L, P, X, I would guess. Or you would have to just remember them. In both cases, you will later on be asked which is the third position. So you would have to, you would be asked about a specific letter and answer in which position that was, either after alphabetizing or after remembering. Yeah. So if it's after remembering, it's in the third position. That is clear. If it's alphabetizing, it would be in the first position. Yeah. So this is how one from the response can distinguish whether people really alphabetize the stuff or remember the stuff. So that's quite elegant. I think it's quite cool. This was one scenario. The other scenario was that essentially the alphabetize test, but with an action required or with no action required to also dissociate motor responses. So that was the task. You can all read about all that in the paper, of course. And then they had a hypothesis in terms of working memory content manipulation, that this should happen in superficial layers of dorsolateral prefrontal cortex. So they only distinguish between superficial and deeper layers, so it doesn't really make sense to talk too much about which layers in terms of this 1 to 6. But the idea is that superficial layers do the processing, and that if you do working memory content manipulation, you do processing, and you need your superficial layers and your recurrent connections specifically, which then leads to the hypothesis that if you are alphabetizing your working memory content in fMRI, you would have ongoing activation in the superficial layers as compared to remembering, because there you don't need to manipulate it, but you just need to keep it in memory. Whereas the action, because the output layers are the lower layers, and that's, I think, here it was a little bit like, because this is a little bit like saying, okay, if you alphabetize, that means that your layers 2 and 3, where all the synapses are, go on. I think to make the hypothesis that the lower, so deeper layers are more associated with motor output in frontal cortex. This is something that is, I think, more established also in terms of neuroanatomical findings. So I believe this hypothesis a little bit more than this hypothesis, but that's just me. So here, if you would need to action planning, then the idea would be, if you're required to make the response and you're only told to make a response after this delay period, then the deeper layers should show more activity if you actually need to plan a motor response than whether you're not doing a motor response. So this is kind of the layer-specific hypotheses that they came up with after seeing the data. They came up in front of the whole thing, registered the whole thing, and then found what is going on. Nevertheless, so I'm a little bit skeptical of this. On the other hand, it's also pushing things, so it's also good. So I don't want to be overly skeptical. Also, I haven't done it, so I have no idea. So this is kind of the neuroanatomical functional work that went into that. So of course, if they want to do layer-specific stuff, they need to be able to dissociate superficial from deeper layers, and the way they do that is to basically rely on the fact that there is kind of this, how do they call it? There's a border or a dip or whatever between superficial and deeper layers, which is due to the fact that there are less cell bodies, which you can see here quite clearly. So you have a lot of cell bodies in superficial layers. So here's superficial, here's superficial, here's I guess lower. Is that the correct? It's not really easy to see. That's it. A dip between layer 3C and layer 4 is taken until the landmark to interpret functional data. Exactly. So you have this dip because there are less cell bodies, and of course, if you now have the functional data, you somehow also need to be able to dissociate the superficial and the deeper layers. And this is done based on this excursion here of the signal. So, and here they just put things next to each other based on histology, of course, in a different and dead participant and their participants. So somehow here, so this is cortex. Somehow, if you do this using if you quantify the signal here, you will also see this kind of thing, and this is where they put the border. Yeah, the whole thing is just about to say, okay, this is superficial versus this is deep. You could also use a classifier for that, and this is a classifier actually, but the classifier here works based on detecting kind of a minimum. And so they can do that in each participant, and to give you a feeling of what the sizes of what we're talking here. So they have this DLPFC, dorsolateral prefrontal cortex area. Essentially, this is superior frontal gyrus. Not gyrus, gyrus. In the different participants, the signal that they then look at refers to what I circle here. So it's quite small. And they have superficial and deeper layers, which is, I guess, not that easy to see here. You only see the superficial layers, but if you go really close, there's one layer in red. Maybe you see it better because you're not as colorblind as I am. There's a layer in red here. So this is the size of it in terms of spatial resolution, because this is what we're talking about it. And now what do they find? Is the question? Yeah.
On the other slide, on the graph. No, that one. Yeah, this one. So where all the graphs are, would the x-axis be spatial component?
Yeah, that's a good question because it's not labeled here. That's not good. They should label it. So this is going from either superficial to deep or from deep to superficial. And I'm not completely clear. One can, I think, get it from these colors. But maybe... what's that? So green seems to be here superficial. So the left on the x-axis must be superficial and then going to deep. Maybe it's the other way around, but the important thing is that it goes from one to the other.
Okay.
And the important thing is here that they know how to distinguish deep from superficial because they find this little inclination, this dip in the different signals.
And these different I don't know, are these different measures, like they measure different signals?
Yeah, this is these are different signals. So this is histology. This is a flash sequence, which is one way to do anatomical imaging. This is MP2 range, which is the typical T1 image. This is functional T1 EPI. So this is T1 weighted EPI, which is not often used. This is bold EPI, which is the EPI that is always used. And this is a new sequence, functional vaso EPI, that this Huber guy invented, which seems to be pretty good to do layer-specific imaging, functional imaging. So this is fairly specialized. So it's really not. This is really novel and specialized. So we haven't discussed all these different forms yet. But T1 and you have heard, and bold you have heard. Now, other questions? No. Now, in terms of their findings, this is what they get from bold. So alpha means alphabetizing condition, REM means remember condition. In the superficial layers, they find that the superficial layers are in fact more active during alphabetizing than in remembering. For the deeper layers, this also seems to be the case in bold. And the deep layer, and the action, non-action seems to not really be distinguishable in bold. So the strongest thing on this graph is definitely here, that the superficial layers distinguish between alphabetizing and remembering in terms of the bold signal, but it seems to be roughly the same in the deeper layers. Things are a little bit more specific, but then when I look at it, also a little bit more noisy from my perspective for this new sequence, which shows a stronger dissociation. So here, if you look at this, not only the bold signal, but this combination of cerebral blood volume and bold, which is given by this vaso sequence, then alphabetizing versus remembering shows strong activity in the superficial layers, and you don't find in the delay period much of a difference here, but things are also more noisy here. In terms of the superficial layers, both of them are active during the delay period where they had to alphabetize. So that's this, and the deep layers are not specific, not shown much here in terms of differences, but a strong difference when people need to do an action versus non-action. So essentially this vaso thing shows exactly what the hypothesis was, which is awesome. And this is kind of if you follow current trends in spatial resolution of fMRI, as you see, this is from last month on bioRxiv, so this is not even reviewed. It's likely that this will come out in a high-profile journal because it's awesome, and the results are so good. So take it with a grain of salt, but at least that's what people are. I don't know, it doesn't like this slide. This is what people are talking about right now in terms of spatial resolution. Yeah? Good. How are we doing? Oh, we're almost done. Then let's stop that here because the next topic would be temporal resolution, and I'm not quite happy to start that. Do you know what bioRxiv is? bioRxiv is a preprint server, so you might not know what preprint servers are. So preprint servers have been around in physics for a long time. This is where physicists and mathematicians put their paper once they are done, and they have done it well. And then these didn't exist in biology or cognitive neuroscience for a long time. That worked that if you were finished with your paper and you did it well, you just submitted it, then it went through review for a year and nobody could see it. And nowadays people also, when they're done with their paper, they publish it on a preprint server where it's not where it hasn't been reviewed yet, and then it's visible, and we can talk about it, and then a year later or so, it's coming out of the review process and is published in a real journal. Important thing about the review process doesn't mean that it's approved and everything has been checked. Review process just meant that 2 reviewers read the paper and gave some random comments to it, and it doesn't. So just a general remark: if you ever review a paper for real, your task as a reviewer is also not to. Your task is, first of all, it's not to decide whether this research is important or not. It was important enough for these people to do it, so you're not in the position to say this is uninteresting. It was interesting to these people, and you're also not in the position to. Your task is also not to, in a way, verify every thing that would into that. Most of the time you might not even have the means, because people are still reluctant to share data and code, so as a reviewer cannot do anything. I think they actually write in this preprint, they write, the data will be shared once the thing is published, which is also something that I find a little bit, this is also what I see sometimes now when I review things. This is also completely ridiculous. I mean, why? You might as well, the only people who actually might be interested in checking things are the reviewers, because they somehow feel obliged, and for them it's not available. That's strange, but yeah, but even if you in the future will have access to actually the data and the code, then of course it's still not the reviewer task to now check everything. That's still the task of the authors, meaning that preprints are essentially as good as real papers. Depends, of course, on how people deal with that. So if people start now uploading things that they think are not quite done yet, but let the reviewers help us to write a paper, and then you find these things on preprint servers, that's of course not good. If people do proper science like they should do, so they finish something, and they finish something that is well done, so they are only done when it's well done, and they can defend it, then preprint papers are as good as review papers, because the reviewers usually the reviewers should only make help to make the communication clearer. Besides that, they don't do much. Good. Any questions review processes or bioarchive? No? Good. Then enjoy your seminar on Thursday and Friday, and we continue with temporal resolution next week in this course.